So we're going to be talking about two main functions of the liver, the ability for the liver to filter and process blood, and then the other function is to produce bile. So here I'm actually going to write the blood pathway in black pen or pencil, and then we're just going to go ahead and bubble that. So it stands out a little bit. We're going to talk about the pathway of blood through the liver first before we talk anything about bile. I did title at Unit 7, Lectures 1 and 2, Liver Anatomy, and usually when I take notes, anytime I abbreviate something, I always write what the abbreviations are in the corner, because you know how sometimes you take notes, you abbreviate things, and when you come back to the notes, you're like, what? Yeah, so my abbreviation for blood, so I'm just going to do like a little key in the corner over here. So my abbreviations. Today, I'm going to use BL for blood. I don't know why I do that, but if you want to use some other abbreviation, feel free. SM is small. And then what else do I abbreviate? Oh, I don't know if this is self-explanatory or not, but that's a heart. And what else do I abbreviate? I think that's it for now. Okay, so the very first structure will connect the small intestine to the liver. In the picture, there is no small intestine, so we're actually going to add the small intestine. The small intestine is located inferior to the liver. If you can draw it, feel free. If not, you can do my simple rendition of the small intestine here. I'm just gonna write small intestine, and then I'm gonna put it in an oval. Ta-da, there's your small intestine. So the small intestine right below the liver is connected to the liver through a blood vessel called the hepatic portal vein. And it is a deoxygenated vein, so I'm going to use a dark blue color pencil for that. In the picture, the printout, you'll notice that it's the vein that's kind of like a darker gray color right there. There is an artery on top of it, so you want to avoid coloring the artery. I'm just going to go ahead and outline the hepatic portal vein. I'm actually going to extend it a little bit farther so it comes a little bit closer to the small intestine. I'm going to color it in. Make sure you avoid the artery that's right on top of the hepatic portal vein. And the hepatic portal vein will actually bring nutrients from the small intestine to the liver. It actually branches off into every single section of the liver. So I'm actually going to draw smaller branches of the hepatic portal vein all throughout to make it look like there is nutrient-rich blood going to every section of the liver.
Isn't it deoxygenated?
Yeah, but it's bringing nutrients directly from the small intestine. Deoxygenated just means no oxygen. Doesn't imply denutriented. Wait, not a word. Just kidding. Okay, and then I'm going to make sure there is a blue hepatic portal vein branch going to every single section of the liver. And I'm going to go ahead and use my black pen or pencil to draw some arrows that show the direction of blood flow. So blood is going from the small intestine to the liver, so I'm going to put an arrow upward towards the liver from the small intestine. And I'm going to go ahead and label that number 1. To the right of the image, I'm going to go ahead and write number 1, hepatic portal vein. So this blood vessel, in black pen or pencil here, I'm going to write the description. This is going to bring blood from the small intestine to the liver. Do you guys want to abbreviate the word liver? To what?
LV. Or LV.
LV, okay. So let's do liver. I'm going to add that to our list of abbreviations. So small intestine to liver, because we'll use that word a few times. So this is going to bring nutrients from the small intestine to the liver. And what's going to happen to the nutrients is, depending on what nutrients we're talking about, they're either going to be stored. So for example, do you guys remember what was it? Carbs, lipids, proteins, minerals, vitamins. Which ones get stored in the liver? Vitamins are going to get stored. Some of the carbs are going to get broken down and used. Some of the proteins are going to get broken down or used as well. And then some of the things in the blood from the small intestine will get filtered. So there's a lot of branching pathways in our drawing here. The hepatic portal vein starts off pretty large, and then it narrows and gets smaller and smaller and smaller as it approaches each section of the liver. And then once it goes into each lobule, it gets even smaller branches. So on your drawing, so find your drawing from yesterday. On this drawing, can you please find your hepatic portal vein? Got it? Found it? Yes? No? Hopefully? Is it in the corner, middle, side? Where are we? It's on the side. Each liver lobule, remember, is a hexagon shape. Each corner of a lobule has the hepatic portal vein bringing blood to it. So in your drawing, do you have arrows from where the hepatic portal vein blood flows? Okay, you should have arrows pointing down towards the center of the lobule, down into the central vein. So the next structure we're going to identify is this path right here, this little freeway for blood. This is called the sinusoid, but you can't see the sinusoid in the picture I just gave you. So what we're going to do on our paper in dark blue, I'm going to write figure 1730 sinusoid. Since we can't actually see it on our handouts, you're going to have to reference your notes every time you want to see where the sinusoids are. So the sinusoids, in black pen here, brings blood from the hepatic portal vein, which was number 1 there, to the central vein. So blood flows from the hepatic portal vein through this path called the sinusoid down the central vein. Think of it almost like a sewage system, okay? Down comes out, down this path, and then down the sewer in the middle. So these sinusoids can be described as small vessels within the liver lobules. Blood must travel through here in order to get filtered out. So you have to make sure that in your drawing, your sinusoids have these little cells inside of them called Kupffer cells. It kind of looks like messed up sunny side up eggs. Do you have those in your drawings? Yes? Do you have them labeled? Okay, good. So in your drawing, you should have these little sunny side up cells. These are called Kupffer cells. Can you please say Kupffer? Kupffer. And these cells, you want to think of them like as stationary Pac-Man. Because you know how Pac-Man in the game, he like runs around the corridors and he's like, num, 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 num. The Kupffer cells stay in the corridor as blood passes. If anything passes that's not supposed to be there, they'll eat them. Okay? So it's kind of like, I don't know, what is that analogous to? Something that eats things as they flow by. The what? Like a fly catcher, yep, as the Venus flytraps. So as the flies fly by, if they land on the fly trap, then the Venus flytraps will shut and eat them. So that's kind of like what the Cooper cells will do too. If it's bacteria or viruses, they'll kind of engulf them and then break them down. Okay. Okay, so those are your sinusoids. Next up is this structure down here, which is the central vein, so which is where all the sinusoids flow down. So I'm going to add number, actually no number, because you can't see it here either. I'm just going to use my dark blue pen and write figure 1730 again. Central vein. And this is going to be blood from the sinusoid down into a larger vein called the hepatic vein. Any guesses as to what HEPA means at this point? We got hepatic vein, hepatic portal vein, hepatic globules, hepatitis. Liver, yeah. So the central veins are found at the center of each lobule. So it's essentially a drain for blood that has been filtered already. Because at this point, once the blood is flowing through the sinusoid, it gets all the bad things pulled out by the Kupffer cells. Then once the blood gets to the central vein, there's really no more things in that blood. There should be no more nutrients. There's no more bacteria, viruses, any other things that are not supposed to be there should have been filtered out already. All of the central veins, so there are actually like millions of these lobules stacked in your liver. Each of them has a central vein. All the central veins merge together, kind of like how all the sewage system from all of our houses eventually merged together at some point in this city. All of the central veins merge together to form the hepatic veins. So I'm going to use my light blue to color in the hepatic veins. So the hepatic veins bring all of the deoxygenated blood that has no more nutrients back to the heart. So using light blue, just make sure there's a light blue branch going from each of the sections of the liver. And this is all deoxygenated blood, no more nutrients, no more bacteria or viruses. This should be nice, clean blood. So this is the hepatic vein. And the hepatic vein actually extends, or it actually starts from below the liver. So this is called the inferior vena cava, which is a really large blood vessel that the hepatic vein dumps into. So this is a part of it as well. So I'm going to use my black pen or pencil to draw some arrows in to point at the direction the blood will flow. This is blood that has no more nutrients, so it doesn't need to be in the liver anymore, so it's actually going to leave the liver. And once it leaves the liver, it actually goes back to the heart. So I'm just going to draw an arrow up out of the liver and back to the heart. And we're going to draw a little label for that one. That's going to be our number 2. So I'm just going to point out one of the hepatic veins, because there are a few hepatic veins here. There's the right one, the central, and the left one. So we're just going to add a number 2 right here for the hepatic vein, one of the hepatic veins. And down below, I'm going to write 2 in light blue and hepatic vein. So number 2, hepatic vein. And that is going to be blood from the liver back to a larger blood vessel called the inferior vena cava, back to the heart. So all of this is filtered blood. So nice, clean, no pathogens, no microorganisms. So this is blood that has been worked on by the liver, and then we'll talk about the blood supply for the liver itself after our stretch break.